Thank you. Sorry I can't be there in person. I would have liked to, but my schedule just didn't allow it. But it's been good to return to more in-person meetings, and I'm jealous that I can't be there. I just want to start with this very just basic concept that we know that our phenotype is a result of a combination of our genes and environment. And this is what has driven much of my work, is that while I focus much more on the environment, I totally appreciate the importance of genes and genomics and genetics. But the idea is it's a combination of the two. And we've had more than a century of great advances on the genomic side. We can get very deep information on the side from multiple tools. It's been really extraordinary improvements in the field. And I think this has been one of the things that's driven this idea of this imbalance that has occurred. So again, if you get back to this idea, is you have this G by E equaling the phenotype, but I draw that E quite small because in our ability to measure it, we're very good on the genomic side, we're very good on the phenotype, but on the environmental side, we really just haven't had a systematic way of doing this. And so this is where this idea of the exposome came in. Chris Wild, who is the director of IARC at the World Health Organization, proposed in 2005 that we develop this exposome concept. And he was a molecular epidemiologist who wanted better exposure assessment. And he defined the exposome as all exposures from conception onwards, including those from lifestyle, diet, and the environment. And many people have simplified this just to meaning the totality of exposures throughout one's life. And the challenge here was that when I started looking at this about in 2010 was when you started talking about the exposome, people would just, that's impossible. You can never measure all of those things. And so one of my colleagues, Dean Jones, and I at Emory, when I was still at Emory University, we proposed a refined definition of the exposome being the cumulative measure of environmental influences and the corresponding biological responses throughout the lifespan. The idea is we measure what we can measure. We can measure persistent chemicals, recent exposures from short-lived chemicals, biological responses as fingerprints of past exposures like epigenetic marks, and really putting less emphasis on that totality aspect and more on the reality of what we're exposed to. And so when we think about the exposome, we're thinking much more than just the sort of chemical exposures like flame retardants and pesticides and things. We're thinking about things like odor and noise and light, temperature, humidity, our ecosystems, access to food and care, green space and blue space, and even things like socioeconomic forces. Like the idea that if structural racism is impacting the risk of cardiovascular disease in a population, it has to alter the biology, right? It's not magic. Whatever's happening there has to be transferred to an internal biological signal. And so the idea is what we're trying to do is to measure as many of these external factors as well as those internal biological signals that are driving it. Our main approach here has been to get as much chemical coverage as possible. And as we heard earlier on the metabolomic side from people like Dave Wishart, who's pioneered much of this work, is our liquid chromatography-based mass spec can pick up large number of molecules. But the more lipophilic, semi-volatile things, a lot of our environmental chemicals aren't picked up as well. And so we found that combining a gas chromatography-based approach with the liquid chromatography gave us much better chemical coverage. And so the idea is we use this combined platform. We use thermo Orbitrap instruments, very high resolution, and do this to try to cover as much ground as we can. And so we can measure untargeted, we get tens of thousands, 100,000 peaks. And that corresponds to many hundreds of things we can identify at a high level. And then a lot that we can't quite identify as well, but we're still getting this very good coverage. And so this idea is we developed this sort of dual platform system, and this has been the approach we have been using. One of the challenges when you look at xenobiotics and environmental chemicals is authentic standards can be scarce. So it's easy to find standards for amino acids and glucose, but if you're looking for a standard of a microbial metabolite that was made in your gut from a pesticide exposure, those standards don't exist. And so it makes it much harder to match up with the various databases that we have to get better identification. And this is one of my concerns here, is that if we don't want perfect to be the enemy of the good here. The idea is that even though some peaks may be unknown, it's not as if they're not unidentifiable. It just takes more work. And so we want to get good identification on as much as we can, and then improve our approaches into identifying those that are currently considered unknowns. And those in the metabolomics are very familiar with Emma Szymanski's work in the Szymanski scale. And this has been very important in getting very good confirmation, striving for those level 1 and level 2. And I like to liken this to describing a car. When we get an exact mass of interest, a level 5, that's like saying I saw a blue sedan driving down the road. A level 4 might say, now I know the make and model, it's a blue Honda Accord. And then when you start getting a bit more data on this, some experimental data, you might say, now I can get the make, model, and trim, like it's a blue Honda Accord EXL hybrid. The level 2 and level 3, level 1 get us to this, I have a license plate number or a vehicle identification number. I'm certain of what that is. And just imagine if somebody had robbed your house and they are driving away in their getaway car, and you could read the first 4 digits of their license plate. Would you tell the police officer that, well, I don't have any information because I only got the first 4 digits of their license plate? Of course not. You would report that partial information that you have because it does narrow the search space. And so the idea here is that as we're looking at unknown environmental drivers of disease, we don't want to limit our search by saying we can only look at things that have reference standards. We have to open it up a bit more if we want to really truly discover novel drivers of disease. One of the challenges that we ran into is that the Szymanski scale is set up very nicely for liquid chromatography, but as more of these gas chromatography high resolution instruments have come along, the scale didn't work as well because there's different things you can pull out of it. And so a consortium of us led by Crystal Pollitt at Yale and others developed this scoring system for kind of a corresponding Szymanski scale for the GC Orbitrap. And this just takes advantage of the different chemical information you can get from that system. And so this is the sort of thing we need to do to get better at measuring those environmental chemicals. We have to adapt our approaches in informatics that allow us to look at exogenous features as well as the endogenous features. And getting back to the sort of xenobiotic idea is, this classical example is we know that the consumption of grapefruit juice can inhibit cytochrome P34A, A4, and this disrupts the levels of many different xenobiotics. And if you think that this chemical found in grapefruit, which is rather innocuous, wouldn't you expect other environmental compounds to influence levels of our P450s and other enzymes? And so the idea is that many of these things we take into our body influence our metabolism. And so this led a collaboration of Dean Jones, Eddie Morgan, Shazal Lee, and I to think about how we could get better at identifying what were somewhat unknown metabolites. And so the idea here, we said, what if we took liver fractions and put them in a 96-well tray and put a known chemical into that vessel, and then took samples every hour or two and ran all those on the mass spec. We know what the parent compound was, we know what's there at time 0, and then things that show up at time 1 hour, 2 hour, 8 hours, whatever after, had to be formed from that reaction. And so we went through and did this systematically on dozens of compounds, both environmental chemicals and other xenobiotics, drug metabolites, and things. And so we went through and did this and found that we could identify many different metabolites of these known compounds, and we would have very exact high resolution mass charge ratios here. And then we could take those parent compounds and inject them into a mouse here in A, and show the formation of the parent compound being absorbed and those metabolites that we had identified in the cell culture system with the exact same mass showing up at the time later. So what we saw in vitro corresponded to what we saw in vivo. And then we could even go into human samples here in G and show that a person that was taking bupropion had those same metabolites that we identified in this sort of system. And so the idea, we can actually do this with mixtures, because if you have compounds that are quite different, you can put in several compounds at the same time and track the development of these various metabolites. And so Shazal Lee he's built a database where he's deposited these compounds in M to Z. And so we have, while it may not be an authentic chemical standard, it is an enzyme-based standard and says that we know this is coming from that compound. And we believe that this sort of mechanism can help us better identify this. We've also now done this not just in the liver fractions, but in cell lines where we've specifically expressed individual enzymes. And so we can attribute that metabolism to a specific enzyme. And this sort of untargeted approach of like, we're looking for whatever we see, we came across a metabolite of caffeine that was nowhere in the literature or databases. And so we suspected that this might be a novel metabolite of caffeine, which is a very highly studied molecule, and went back and did stable isotope labeling, and sure enough showed that there's a new metabolite of caffeine that was found by this untargeted approach that required follow-up and identification. But the idea now is that this novel caffeine metabolite can now be put into a database for future matching, even though there's not a true authentic standard. So now we've taken these sorts of approaches into various human studies. This was an early study that we did with the Mayo Clinic looking at primary sclerosing cholangitis. This was a pilot study that we did that led to funding for us now to increase the N to 800 per group. This is only 40 per group that we showed. But the idea is here we can measure these different environmental chemicals and show in a network plot how they correspond to disruptions in various biological pathways. And again, this was an example where we identified some alterations in bile acid metabolism that the group had previously not seen while they were studying this liver disease. And the idea, it's this untargeted approach that captures the environmental chemicals and these endogenous metabolites. And so now in this current study, it's a multi-omic approach. We're doing fecal metabolomics, plasma metabolomics, transcriptomics, proteomics, very comprehensive multi-omic approach here. And this is the sort of thing that we think can help connect those exposures to various outcomes. I was also very interested in looking at some neurodegeneration factors. This was an early study that I did when I was still at Emory. And like other groups had reported elevations in glutamine and some other compounds. But I was very excited by this observation that we found this metabolite of DDE, which is a metabolite of the pesticide DDT, that it was elevated in the Alzheimer's patients. And I was especially excited by this because a former postdoc of mine had done an analysis, a targeted analysis of pesticides in Alzheimer's disease a couple years earlier in a different population and had found a DDT metabolite. So again, we're doing this in an untargeted fashion, looking for anything. And there was literature confirmation that some of these things had been found in similar populations. And so this kind of gave a clue that this sort of approach in this untargeted way could be helpful. And again, this was a relatively small study, about 120 patients or so, relatively small. But it shows that we can detect the drug metabolites, dietary factors, and environmental chemicals. So then I moved to Columbia a few years ago and started collaborating with Richard Mayhew, who's chair of neurology, and he runs this very large project, Washington Heights and Inwood Community Aging Project. And here we could show that we could separate cases from controls, but could also separate the ethnic groups, which are otherwise not different. There's no genetic differences. Everything else they looked at, they hadn't seen differences, but this very detailed biochemical analysis could show these differences. And so this was a pilot study we did. We then got a grant to run this on over 3,000 participants. And when we look at the metabolic changes that occurred, we see differences in the different ethnic groups, which is important because there's differential vulnerability to the cognitive decline. And so this starts giving us some ideas of the pathways that may be disrupted that lead to these disparities we see in the health outcomes. And so the project is now underway. We've probably run well over 1,000 of these samples, but we see the incident cases. We have multiple measures in this longitudinal design, and we are very carefully doing this analysis now and hope to have results in the next few months. Based on the finding that the Caribbean Hispanics in New York had a higher incidence, Richard started a study in the Dominican Republic. And this was exciting because in addition to doing the plasma analysis, we could also get cerebral spinal fluid and look at the Alzheimer's protein biomarkers and look at these connections between what we saw in the cerebral spinal fluid and in the plasma. And so again, in this study, we have some very interesting hits. It's too early for me to talk about it, but it's some very interesting things, and it's nice to have this connection to the YCAP cohort as confirmation. So other groups have been studying the populations up here in northern Manhattan, looking at air pollution and these cognitive decline outcomes. And so one of my postdocs said, well, if we have data on the air pollution, and I have data from the metabolomics and exposomics, and we know their addresses, can't we map the levels of pollution onto these metabolic changes? And this is, I actually took the slide out. And so we actually did this and could show that there were features in the metabolomic analysis that corresponded to the levels of particulate matter 2.5. So merging those exogenous measures of pollution into the individuals that had increased incidence of Alzheimer's disease. And so it really helps you kind of bring in the external to the internal. This last example here, looking at Parkinson's disease, we did an initial study of patients that were being treated and found that most of the changes we saw were due to the L-DOPA levels into their bloodstream. It changes a lot of biochemistry, and knew we had to go to a more naive population. So we obtained samples from the European EPIC cohort, which is a cancer cohort of 500,000 people. Hundreds of them developed PD. We got blood samples from when they entered the study, average 8 years before diagnosis. And we have run this analysis, and we've seen some very interesting things, and we're doing the follow-up on it. But the idea of like finding these appropriate cohorts and these biobank samples let us tap into these things very readily. We can also bring these things back into the laboratory. We can look at various things, modifications in C. elegans, do the high resolution mass spectrometry, and look at the changes in the biochemistry that correspond to the RNA-seq and things that we see with this. So the idea is it's not just an observational thing in humans. You can bring this back into mouse studies, C. elegans, even Drosophila, to do various G by E follow-up. And this is an example of this. Following up from our Alzheimer's study, we were able to look at mutant worms that had tau levels, expose them to that DDT metabolite, and then look at how that was affecting various biological outcomes in the C. elegans. And so we think it's a really good tool to go back and forth from the human populations to these biological systems. Another example, just thinking about this, the theme of this meeting, I recalled this paper we did a couple of years ago where we developed these microneedle patches that could sample interstitial fluid, just like kind of like wearing like a Band-Aid. They're not ready for widespread use, but we've shown conceptually that it works. But this gives an opportunity to see a way of how you can collect biological samples without someone going to a phlebotomist or a doctor's office, and democratizing the collection of biological fluids so that we can do these sorts of mass spectrometry techniques. And for example, the NIH All of Us program, which will have over 1 million participants, is now more interested in looking at environmental things, and we're trying to see how we can bring exposomics into this. And I think this is an exciting opportunity. The examples we've seen from UK Biobank, when you can go to scale, you get very good statistical power. So to me, a key part here is really scaling this, is we want to look at all these tens of thousands of features in an untargeted and hybrid approach. But it would also be great to get information on hundreds of environmental compounds in diverse cohorts. And to me, like having like an exposome kit that you could measure, 400 or 500 compounds at level 1 would be great. And this is something I've talked to some of the team at Biocrates about what it would take to do this. The challenge is that we have hundreds of thousands of chemicals to start with. How do you pick those 400 or 500? And I think this is something that would be very helpful in advancing our understanding of environmental compounds, but it would take quite a bit of effort. So with that, I'd like to thank my colleagues, and I'd be happy to take any questions.